Okay, so that we were here, right? The real world data. We were at okay. So how do we experimentally figure out where each of the histone modification or any of the transcription factor or any of the DNA binding factor, that where are they, right? That's really the goal for the next hour or so, next hour and a half, next hour, 2:30 to 4. I was supposed to finish at 4. Let's see how long we take, okay? Um, so why does this matter? This matters because of all the patterns we talked about earlier, right? This matters because we want to understand where an active gene is, where a repressed gene is, where. So, okay, let's take a step back. Let's think about this. Okay, lots of people are missing. Is it true? Or am I imagining this? All right. The people like to really sleep after eating, right? Don't they? Most of you go home and sleep, huh? Yes? No? How many of you sleep after lunch? Nobody? Sure? Okay, can you guys move up then? The people who are in the back? What? Are you feeling hot there? Don't know why you have this phase and you have that phase. And so on and so forth. You can't know this. No? You can't know this because you have the technology. The technology just developed 20 years ago. Actually, the technology was developed in University of Cambridge by a guy called Shankar Subramanian. Indian, brilliant Indians outside of India, right? Inside. We're talking about Dr. Rahman and Murthy, IIT, done a lot of X-ray crystallography work, amazing work. Lots of great people, but people around nephew, molecular biology, really, really good people out there. I'm not really being a racist, but they're really good people. Just do a lot of good things. Venki Ramakrishnan, we talked about, right? Dr. Kalachudman might get a Nobel Prize one day for discovering sequencing technology. One of them. The other is the state mentor was like the one thing that I haven't had state mentor. The basic venture capitalists who actually invested in doing sequencing or genome or sequencing. So now, by adaptation of simply genome-wide sequencing, you can sequence a lot of stuff. You can sequence RNA, RNA-seq, you can sequence epigenome, lots of epigenome sequencing methodology. That's what we'll be talking about, okay? That's what I specialize in. My specialty is in epigenome sequence. That's why I love this stuff. That's why I'm spending 2 hours doing this. And I'm like, we talk about the rest later, right? No, because that's important. That's important for you to discover new things. How do you discover something new? If I was just going on studying alcohol, I written it, I'll make some more discoveries, but it's just being incremental. But to make something totally new discovery, I need to look at things with an open eyes, like somebody who's never done it, right? So I believe at some point in my life, I get a good thing with discovery-based approach. CRISPR-Cas, we talked about Levi Garraway's stuff, right? CRISPR-Cas, he didn't discover it, for example, right? By doing by genome-wide CRISPR-Cas-based knockout screens, people can discover new things that nobody never knew. They never knew MTAP deletion methods could be different from PRMP5. New discovery, new therapy, new hope for the patients, right? Same thing. New biology is discovered by being unbiased. I realized this in year 2008 when I was publishing one of my first DNA democration papers that, wow, I could study one enzyme or I could discover many new things by just looking unbiased discovery, right? So that's what we do. I take human patient samples from clinic to come to ambient. We take them, we sequence them, and we make maps like the one I showed you earlier. Epigenome mapping is where my lab has become famous for what we do. Okay? Because we want to understand how this code works, right? So here, let's take a simplified example. We talked about HTK4 methylation. Where does it stand? Which all these have the potential to be activated, which all these are suppressed. How is in a different cancer, a cancer that affects, let's say, retina? What happens? What changes? We don't know. We just know there's cancer. We just know. So much of cancer biology is not because we didn't know. It's never looked. Right? So much of diabetes biology is unknown because we don't know how beta cells function out of suppress insulin. We know a little bit. Diabetes is relatively easier to use compared to something like cancer. Relatively, not under my name. My bottom line is, because we don't know, we should look unbiasedly in things and discover new things. That's what we do. So, the histone modification, right? It should be specific to a given locus, right? That's what we looked at. Or it could be across the locus, like multiple places, right? So it could be widespread, it could be at a specific position, it could be unbiasedly there. Not working. So many genomic locations, right? So which genes or genomic regions are associated with a specific modification of histone or a specific transcription factor or a specific DNA binding factor of any kind, right? All you need to know where this thing is binding in my genome is done by these methodologies, okay? We'll talk about these. There are 3 major methodologies that are used right now. There are others out there. ChIP-seq is what I'll tell you about. We do tons of ChIP-seq in my lab, so I'm like married to ChIP-seq, right? But there is a new technology called cut and run that works much better actually these days and works in small number of cells and is much cheaper, so people use that, and a version of it called cut and tag. I'll only talk briefly about cut and run later because again, I have to talk about what I'm married to, right? So ChIP-seq. ChIP-seq's been an old technology, works well, but the concepts are pretty similar. So once you get one, you can easily adapt to the other one. Okay. What is ChIP-seq mean? ChIP-seq stands for chromatin immunoprecipitation. Okay? Just don't chip seek. This is Arpan's slide, so I'm just, pulling it. Okay. What does immunoprecipitation mean? How many of you know that? 2, only 2, 3. That's it? 4. Okay, one of you, you're gonna come up, stand, and tell me what immunoprecipitation mean, or tell your friends.
Protein recognition by the antibody, and antibody is precipitated by the using magnetic fields or icons.
He's my epigenomics guru, right? And what does immuno mean? Immuno is referring to the antibody, use of an antibody, because antibody is equivalent to immune system, isn't it? Well, not really, but sure. Okay. So immuno means it's referring to the use of antibody. Precipitation is pulling something down. So chromatin is chromatin. So we are using an antibody against chromatin to pull it down. Very simple. Seek means seek, sequencing it. I talked to you about sequencing, right? How many of you have heard of next generation sequencing? Everybody. Has a teacher taught you that? You guys don't know sequencing. Okay. We have a long curve ahead of ourselves then. What does sequencing mean is, okay, I probably have a slide. We want to go to next generation sequencing first, because I don't want my other friends to feel they don't know what we're talking about. Oh, this will take a while. This is simple. Yeah, here you go. Okay, you guys understand DNA? Understand RNA, right? You can take any DNA, for example. Do you guys know how to isolate DNA? How many of you isolated DNA? Everybody's isolated DNA? Except you haven't isolated DNA ever. You don't understand DNA. What do you do? I'm sorry. Huh? I can't hear you. Pharmaceutical department. That's why. Okay. Okay. So when you can isolate DNA from a cell, right? You just use different kinds of buffers and you isolate DNA. So you can read about it. Okay. Anyways, we can de-isolate DNA, right? We use buffer P1, buffer P2, buffer N3 from Qiagen kit. Remember, I know my shit, right? And then you precipitate it, put it on a Qiagen column, and you take it out. How many of you have done that? Well, very few of you. Okay. You can do it without Qiagen column too. Just use buffer P1, buffer P2, buffer N3, and then isopropanol precipitation, pull it down. Or you can do phenol chloroform extraction, right? All of you know that. Okay, you can isolate DNA. You can read about it, right? Watch a YouTube video on it. I am promoting YouTube a lot. Sundar Pichai is one of my seniors from IIT Kharagpur, right? He was 7 years senior to me. He just had $1 billion earning, I think. I was like, oh my goodness, what am I doing? All right. Another guy, another colleague, another friend of mine was a CTO in Flipkart. He made a company called Udan. His net worth at few years ago was 11,000 crores. And I was like, what am I doing in my life? Friend next door, neighbor in IIT. Anyways, I'm just kidding. But basically, there is DNA, right? You can fragment it. You can fragment DNA by cutting with restriction enzymes or sonication. Have any of you heard of sonication? Sonication is where you put the sonic waves and that kills the DNA, breaks the DNA apart in random pieces. You know this? Most of you know this? Okay, good. Yes? Sure? Okay. So you can break the DNA and then you can add small pieces of known DNA, which is called adapter, through an enzyme called T4-DNA ligase. You can ligate it to your thing. Okay? So you take a piece of DNA, You break it apart into small pieces. You put two pieces next to it by ligating pieces of DNA to it. That's called ligation. So now you have taken your DNA, you put two pieces of known sequence to it. So now you can design primers against that known sequence, and you can do something called PCR. How many of you have heard of PCR? Everybody knows PCR. How cool is that? How many of you have done PCR? Ah, man, this is a sad story. Not everybody has done PCR. Polycomb, polymerase chain reaction. Everybody knows the theory of it, I'm sure. Everybody reads books. We all read books. How many of you really done it? Half of you, 50%. Okay. So master students have not done it. PhD students have done it. Is it? Sorry, what? PCR is an amazing discovery. Unfortunately, it never got a Nobel Prize. Right? Polymerase chain reaction. Very simple. Put a DNA polymerase, put some DNTPs, put some in buffers, and allow this to go through thermal cycle, denature your primers, and go denature your template, let the PCR go through, the polymerase will work, it will make a lot of DNA, right? I know my shit, right? That's what I say. I know, I've done lots and lots of experiments, guys. These days, I only sit in my office and send reply to emails. This is what happens. Okay, good. That's lovely. So, did you guys get it? Piece the DNA, put the two ligation, PCR chain reaction. PCR is polymerase chain reaction. We do PCR. In the case of sequencing, we do something called bridge amplification. That's where the brilliance of these people was. Bridge amplification, where your primers are attached to a chip. Okay, so basically polymerase goes like this and chips, and then you add fluorescent DNA, fluorescent NTPs every single time, and the fluorescence is read by the machine. So the fluorescent nucleotides are detected, and we know which fluorescence is attached to which nucleotide, and we know what fluorescence came what next to what, so I can sequence the whole DNA. Oh my God, so cool, right? We could sequence the whole DNA into pieces. The pieces were sequenced, then they were assembled because we have a pool of DNA that is randomly sonicated, so you can assemble it over each other, right? Did you get this? That's our computation process. That's where your bioinformatics comes into play, right? So this was what? Sequencing by synthesis. That's what SBS, that's a technology, right? So the Illumina has now monopolized the technology. Then the new machines have come, and the new machines have come, and the new machines have come, and each machine costs several $100,000 and makes a lot of money. Another way to become billionaire, right? Illumina is one of the most successful companies by just sequencing, because people want to sequence. They want to know. You want to know, right? You want to know what's wrong with me, right? I want to sequence my genome. Have you guys heard of a company called 23andMe? Nobody. Oh, man. Nobody's heard of a company called 23andMe. They sequence people's genome. You can send your DNA, they'll sequence it, and they'll give you back the data and tell you, you might have a risk of diabetes tomorrow because you've got these alleles. 23andMe just went bankrupt, unfortunately. But it's a great service to the nation, day service to the world, and such companies exist in India too. Lots of such companies. One of my seniors made a company called Map My Genome. Her name is Anu Acharya. You can look, it is in Bangalore. Okay? What did I say? You can sequence things by using a technology called SBS. Now people get it? Are we on the same page? So now given we have sequencing, and given we have ability to pull down specific pieces of DNA of any kind, I can marry the two in, sequence, like pull it down, sequence it, that's it. That's the magic, right? If I can pull down specifically, that's the key, right? If I can specifically pull down where my histone H3K4 trimethylation is present through use of an antibody. Antibody is the best thing. because antibody is the specificity, very, very specific to its target, right? Do you guys know how antibodies work? How many of you need an immunology lesson from me, from a non-immunologist? Do you guys know how antibodies work? Who said yes? Raise your hand. Okay, you want to stand up and explain to everybody how antibodies work. There are people who don't know this, right? There are people who don't know this, right? Okay, go ahead.
They are Y-shaped structures. They have constant regions and variable regions. Those variable regions are actually modifiable, and they are directly complementary to the epitopes of the antigen. They go and bind to those epitopes, and ultimately that complementarity leads to inactivation of the antigen, and that clumping or antigen antibody binding is the actual interaction that occurs in immunotherapy.
Great. The whole key thing in this is antigen. What does antigen mean? Okay, somebody else. Thank you. What does an antigen mean? Any protein, any chemical against which an antibody can be raised. What cells in make antibodies in our immune system? B lymphocytes, not any lymphocyte. T lymphocytes kill things. They're killers. They're helpers. They're killers. But B lymphocytes make antibodies. How do they make specific antibody to specific antigens? Looks like you do immunology research, right? Okay, I'm not an immunologist by training, but I know somewhat. So good. Antigens is the thing. If you have specific antigen, you have specific antibodies made against it. You can do this artificially by putting the antigen in a mouse or a rabbit, and they'll make your antibody, right? Again, and lots of companies do that and make a lot of money out of it. Not a $1 billion company, but several $100 million company. All right, we could do that, right? So you can actually, what is it called? Immunize the mice, immunize the rabbit with the antigen. So you essentially make antigen peptides, synthesize it, inject it in rabbits, and let them do the magic. And then you take their blood out, you filter it out, you again specifically use your antigen to pull down the antibody, you got your specific antibody. That's how companies make antibodies. And you can make lots of money, right? Because there are lots of uses of antibodies. So antibody is specifically is used in chip or cut and run or whatever experiment, because I can use it to pull down what I want, right? That's my chromatin immunoprecipitation. So here's what he shows here is a specific piece of DNA that has got a specific modification. In this case, methylation, could be acetylation, could be ubiquitination, I don't care. It is something. He uses a peptide, a histone H3 peptide, N-terminal peptide that is specifically modified. Immunize the mice. You can get your antibody that is specific to H3K4. Now I want to pull down H3K4. So I take my cells from which I want to pull it down. I make an extract in a buffer where I would have just the chromatin prepped out of it. So how do I prep chromatin? How many of you know that? What is a whole cell extract? Anybody knows what extract means? You guys don't know what extracts mean? How many of you done Western blots in your life? You've done Western blot. What do you do in Western blot? You make whole cell extract? Huh? You don't understand what is whole cell extract?
Entire the things in the cytoplasm and nucleoplasm are.
How do you do Western blot? Exactly. How do you isolate the protein? Huh? There was a kit. This is the magic that everybody does these days. That's the problem, guys. What's the point of following a protocol if you don't know what the protocol does? This is what I tell every single PhD student in my lab. Because there are kits available, boom, I do kits and kit gives me what I want. What's most important is to understand how does kit give me what I want. Okay? Get it? So next time you do an experiment, figure out how kit does it. Okay? That's the key, guys. I take my cell, I take the cell membrane out, so you can sit down, right? That is where I make my whole cell extract, where I make chromatin prep out of my whole cell extract. How do I do that? I use specific sets of buffers. My buffers contain specific sets of detergents and detergents. What is a detergent? Who knows what is a detergent? That's with which I wash my clothes. They break down specific interactions, right? Because they make things easy for you to take out, whatever that means at this point, huh? Break down cell membranes. You use a high salt to break down interactions, and you isolate. Then you do a centrifugation step. You get your membranes out, then what's you remaining is whole cell extract of sorts. Go read about how to make proteins, whole cell extract, right? Please do. Okay. If you don't understand these simple things, how are you going to be a good experimentalist? तुम लोग लैब करते हो कि नहीं? तो लैब करते तुम्हारे साइंटिस्ट जो भी लैब कराते हैं तुमको बता देंगे नहीं प्रोटोकॉल में क्या होता है, कि बोलते हैं जाके किट यूज कर लो, हैं? समझाते हैं कि नहीं? We are on YouTube, guys. Please make sure you respond responsibly. Okay. Bottom line, please try and understand every step of a protocol. First, remove the cell membrane. Then you use buffers that contain sodium chloride or potassium chloride or lithium chloride, depends on the what buffer it is. Then you use a detergent. You use EDTA. What is EDTA? Chelating agent that takes care of. What does EDTA do? Let me see. I think there are 5 different views here. Absolutely. The ions are trapped in the EDTA because it's a ring-like structure. I don't know what it is called now. But anyways, it traps the... Why do we need to do that? Enzymes that are proteinases, etc. Okay. All right. Good. So you make a buffer. You then use a proteinase inhibitor. Then you make your lysate, right? Do you guys do this or no? Good job. All right. That's exactly the same way you make a lysate on a nuclear extract. You use buffer A, buffer B, buffer C, and then you make your nuclear extract, where you would have taken out all the cytoplasm and you would have just got a nucleus because chromatin is in the nucleus, right? That's you make your nuclear extract, okay? And then you put it over my antibody. My antibody can be conjugated to a magnetic bead or a Sephiroth bead. Have you guys heard of any of this? Good, you have, because you're an epigenetics guy. Who else understands what Sephiroth beads and magnetic beads mean? No? Okay. So then I also don't remember all the details. But basically, these are beads that have open pores and specific charges. So you can conjugate your antibody to the bead. The bead can be pulled down. A magnetic bead could be pulled down with a magnet. So in a test tube, your magnetic beads can be on one side and you can take out everything else. So it helps you remove the reagents all the time. That's magnetic beads, okay? Sephiroth beads can be used on a small centrifugation. If you use 2000 G or 200 G or something, then you can pull down specific beads separately so you can take out the rest of the solution. So your beads can be, the solution can be exchanged essentially. That's what you're trying to do. You get this or no? Okay, good. So you essentially conjugate your bead to your antibody, right? A bead can be of any kind. You can use, as I said, you can use open these beads or magnetic beads or whatever. And then you conjugate to the antibody. Then you incubate your lysate. So you essentially put the beads with antibody with your lysate together. Okay, that will help you pull down things. Okay, that's, I just explained this in words. It's all here. Don't worry about this for now. Sequencing antibody and sorry, pull down using a specific antibody, specific piece of DNA, and then sequence it. That's the overall view. Okay, and you're gonna get tracks like this that I showed you already, pictures like this, which is where your epigenome is. So that's the overall summary. We're gonna go deep into steps, right? First, you do antibody incubation against your whatever your thing is. Okay, so you make your lysate, you do your incubation. Actually, he's written it opposite. It's sonication first, then beats, then antibody. Let me see. Yes. Okay. So this is the correct way. Okay. He's wrote it opposite. So you make your lysate, you sonicate it. Okay, that's my slide. I'm sorry, that's Arpan's slide, but I think he's wrong. You have your DNA, right? So your DNA is like this. You fragment your DNA, essentially you sonicate it. Sonicate helps fragment. You can use an enzyme also to fragment, and then you immunoprecipitate. So you put your antibody, now you incubate your lysate, as I said, the sonicated stuff with your antibody. Antibody will pull down. So you take your beads, you pull down the DNA, you wash it off, then you remove everything else. So you pull down means you incubate your sonicated extract with your antibody and beads. You can go reverse. You can actually just incubate with antibody and then pull down the whole thing, or you can first conjugate the antibody with the beads and then pull that down. Either one works. Okay. And you pull it down. Then once you've incubated like for 4 hours to overnight in the cold, you take it out and you release your DNA by just using proteinase K. So that takes care of all of this. Now you have DNA, you sequence like what I told you earlier. Now you've got your sequence, your DNA that you wanted. You sequence with the way you prepare the sequencing library, sequence, and do the computational analysis. That's the overview. You get this? How many of you don't get it? Because I can go back to this again. Got it, everybody? Okay, very simple. Okay, nothing big deal. All you need to do is specifically going stuff. Okay. So I think Arpan had got it opposite, which I'm sorry. So you get your extract. You say step 1 is sonication, step 2 is incubation. Okay. Then you pull down your chromatin essentially with the beads, pull down here with the beads. Then you release your fragments, you sequence it, and then you go sequencing, you get the sequence reads like this. Okay, so now next step. Once you get your sequence out of the machine, that sequence, the PCR'd it up, remember, and sequenced it, it gives you the reads. Meaning in a computational file, you've got what sequence looks like. Okay, so what my 50 base pair sequence looks like, could have 80 GC data, GGC, whatever. Okay, 50-50 base pair like that, right? You got your sequence. That's called your reads, sequencing reads. You align it to the genome. Do you understand what that means? So you have a computational tool. You have got the whole human genome, right? It's called reference genome. Again, we only had a few of them earlier. Now we have 10,000 of them, or we have a lot more of them as we go along. A human reference from India could be slightly different from a reference from America, but people just consider all of them as one human reference at this point. They sequence them, they align it to that to say, okay, where my read matching is. So you read a 50 base pair that's going to be unique to that place. Okay, so it's going to find where my unique matching is. Some places, sometimes you find DNA that is in the repeat elements. What does repeat mean? Same sequence again and again. So you might find sequence breeds that are unique to that repeat. It's not unique, they're actually matched to multiple places in the genome because they're in repeats. You remove them, you don't use them. Okay? You don't use them because you don't know with confidence where they came from, because there are multiple locations that they could have come from, right? You guys got that or no? Good? Okay. We'll come to that. I'll talk about computational analysis. For those who are interested, others can sleep during that time. Okay. Then you do peak calling. So when you peak, your reads align to a certain place. They repeatedly align to that place. That means the modification was actually present there. That's the peak we looked at when I showed you earlier the data. Okay. How many of you are sleepy? And that's what you saw earlier, histone modification. Peak. Okay, histone ChIP-seq peak. That's not K9 trimethyl, that's K4 trimethyl. We know that. That's a typo. Okay, cool. Got this? And then you can have it at different places, different results, and you could have made the map that I showed you, this kind of map. So essentially, we just did this. We now did a K27 acetylation ChIP-seq, we did a K4 mono ChIP-seq, we did a K4 trimethyl ChIP-seq, and a K79, and a K27 trimethyl, and a K9 trimethyl from the same sample, right? So I can now put them together and say, boom, we got this here and here and here. Sorry, 1 second. Okay. Did you guys get this again? I can go back to this because this is the whole crux of the thing chip seeking process. Yes? Yes? No confusion? Nobody's confused? You're not going to raise your hand tomorrow when I ask you if you're confused? Sure? Okay. We'll go to back this again because I just do have to. The ChIP-seq can be used for various things. Histone modifications, transcription factors, histone variants. We refer to that. We'll talk about that in a day or two. RNA polymerase. I can actually chip for specific phosphorylated forms of, RNA polymerase. Remember, I told you that RNA polymerase specifically gets phosphorylated when it's an initiation versus elongation versus pausing. So in fact, indeed, specific residues on RNA polymerase C-terminal tails are modified and there are antibodies to those. So you can use it. So you can say, my RNA polymerase is firing, but it is not elongating. But now this RNA polymerase here is elongating. So I can learn the states of transcription by just looking at RNA polymerase ChIP-seq status, phosphorylation status ChIP-seq. Okay, is that cool? Did you get that? So you can do a lot of things. You can be creative. You can essentially ChIP-seq anything that binds to DNA firmly. Because if it doesn't bind to things firmly, then it won't be. Let me elaborate on that for a second. Everything in your body, every protein that binds to stuff binds with a specific on and off rate. You guys know what does on and off rate mean? No? How many people don't understand that? Most of you don't understand on and off rate. Okay. An enzyme binds to its substrate, right? It has to bind to its substrate to do its job. It will be there, it will have a certain affinity to its substrate, which is measured in terms of KD. Have you guys heard of anything called dissociation constant? Now everybody knows what I'm talking about, right? Dissociation constant refers to how tightly something binds to something. A binds to B, right? Same way, any DNA binding protein binds to DNA with a certain dissociation constant. That depends on the charge interactions between, again, everything is interaction between a pocket and a, that's the same thing. So if you have something binding tightly, means the electrostatic or the covalent interactions are strong enough for it to stay there for a long time. Then only you're going to be able to do it, right? If something is coming in, off, coming in, coming off, coming in, coming off, you're not going to be able to chip that thing. Does that make sense? That's what I said. I said if something has a high on and off rate, on and off rate, very high, means very quick, then you're not going to be able to chip that, right? That's a limitation of this, okay? So you need to have strong enough tie binding constant or whatever, that it has to be there for enough time for it to be able to chip. Okay, got it? All right, good. So we got this. Again, repeating it, DNA bound protein, sonicate it, pull down with antibody, incubate it. With that incubation, pull it down, use proteinase K to release it. You got your DNA. You prepare a sequencing library like we talked earlier. You take adapter, you ligate it to that preparing library. Got it. Okay. And then we do computational analysis. Here is a visualization for those of you who didn't get it, but this will make it clear. Here is our genome. Our genome has things in it, right? So we have different modifications essentially, all right? Different modifications at different tails. So we do something called cross-linking first. I missed that step to you guys. Why did I miss it? Because it was not there. Cross-linking is needed in chip. In cut and run, we don't do cross-linking. Cross-linking is needed because your protein that is on the DNA, so your nucleosome there, stays there. So let's say, so you essentially are ensuring that your protein of interest, your transcription factor or your histone modification or whatever is actually bound to the DNA. Wherever it is binding, it just, we're making it stronger, simply, okay? By just cross-linking. What does cross-linking mean?
Link it to different molecules
Together. We use